Good morning, everyone. As a part of this GIAN course, we have an exciting lecture series, and we have an invited professor, and he's Dr. Mayappan. He's an assistant professor in Department of Biotechnology, IIT Madras. His research includes systematic bioprocess development for biopharmaceutical and cultivated meat manufacturing, data-driven cell line development for both biopharmaceutical manufacturing and multi-omics data integration, and using constraint-based modeling algorithms for this. In terms of plant metabolism, he has previously developed the first ever combined metabolic regulatory network of rice plant and subsequently used it for characterizing cellular behavior in two different tissue types, including germinating seeds and photorespiratory leaves. In addition, he has also led the development of first ever rice genome scale model and utilized it in combination with multi-omics data to characterize the transcription regulation of cellular metabolism with respect to different colored light sources. In terms of covering plant secondary metabolism, he has also contributed to sequencing and annotation of Panax ginseng, medicinal plant which is known to produce a wide array of ginsenocytes, and also led the development of genome scale metabolic model of pan ginseng. And recently, he's also contributed to functional elucidation of various formulation of medicinal herb ashwagandha to multi-omics approaches. So we are glad to hear you on this.
Thank you, Samita. Thank you for the detailed introduction.
Yeah. And as a small token of appreciation, we would like to present a small memento on behalf of the organizing team. I request Professor Smita to kindly present the memento.
Thank you. Thanks. Okay. Good morning, everyone. Thanks, Samita, for the kind introduction. So currently, my research, even though does not focus too much on plant cell, but I had done my previously the PhD about 10 years ago, completely on plant cells, focusing on how rice adapt to various stresses, including the abiotic as well as biotic stresses. And I used computational approaches, particularly integrative multiomics as well as the computational modeling approaches. So I'll try to share about that, and then later I also contributed to the genome annotation of Korean ginseng, which is one of the herb which has multiple health benefit effects, which I would also touch upon. So to start with, I'll just give a context on what about rice. You all know that it's a major food consumed in the world, and most of them they are actually consumed in Asia, as you could imagine. But surprisingly, you could also see that rice is also consumed widely in Africa as well as parts of South America. So this shows that the increased consumption of rice as well as throughout the world. And where they are grown, they are also grown worldwide. So the main thing is, again, Asia produces most of the rice, but then again, different parts of the world, including US and other South American countries, they also contribute to a lot of rice manufacturing. But the thing is, even though the rice production has increased over several years, through, particularly after the Green Revolution, but then the continuous increase of human population as well as the sustained challenges posed by the climatic changes, there will be a point at which the production of rice may not still be able to meet the demand. So that's what this graph actually shows about. And then as I mentioned, the population of the world is also increasing. So there could be a meeting point where the demand actually meets the exact requirements. So because of that reason, we need to understand how we can improve the rice production. So actually, this is a cartoon showing the rice plants, and it is subjected to various stresses. The first and foremost, it could experience this drought stress. What's a drought stress? Drought stress contributes to the water scarcity, and what impact it will have is, because there is a lack of water, the photosynthesis will be reduced, and it will also produce excess amount of toxic reactive oxygen species, which is mainly because of the oxidative mechanism it will try to use. Then the other extreme, countries such as Southeast Asian countries or even India and southern India mostly, they also go through a periods where they will be submerged. Rice plants will be submerged throughout. So this causes a flooding stress. So what a flooding stress in terms of plants, how they experience it. So when there is a waterlogging, the ability to dissolve oxygen into the water is very low. The solubility of oxygen is very low. Because of that, the plants will not be able to get sufficient oxygen, and then they will be experiencing the stress as well. And then the third kind of stress, which is also experienced by multiple cultivars of rice, is the soil stress, which is when there is a seawater mix or increase the salinity in the soil, then there will be a osmotic environment built up around the root, and then this will ultimately decrease the nutrient consumption. And the light also, the different type of light also play a role with the changes happening in the ozone layers and the UV radiation and so on. The different type of different colored lights, the intensity and the wavelength of the lights, how it is being perceived by the plants, they will also have a significant impact. So the last one is the biotic stress. So when rice plants are grow, they also co-host several microbes. One of the common microbe which affects rice plant is the Xanthomonas oryzae. So which causes a rice leaf blight disease. So it just manifests into the rice, and then it tries to take away all the different photosynthetic parts of the rice. So because of this, then because they are endophytes in rice, they will not be able to utilize their nutrients, whatever they are able to produce for the rice plant to grow, and the seed germination and other processes would be delayed as such. So the approach which we wanted to adapt in order to solve all these multiple problems in this next 30-40 minutes, what I'm going to cover is about how we can use or characterize what's happening in these stress conditions in rice plants and how we can overcome that. So it could be multiple approaches. We can try to come up with transgenic varieties which can be more resistant to these stresses, or we can even try to think about making sure the conditions are not as diverse as it is. For example, with the concepts like urban farming, you will be able to try to control the environment where you will be able to get the most productive environment out which can be designed in a controlled manner. Okay, so I think maybe I can just quickly go through this. So this is just to give some kind of glimpse of what happened in rice. So the snapshot is back at 2014 because I defended my thesis in 2014. So after that, I have not had a chance to update the slides. So it all started with the domestication of rice. The first ever probable research which has gone through in rice is when we knew the art of growing them and the controlled environment. That would be the first possible research, as well as identifying the different varieties suitable for different lands and what is the optimal cultivation periods, harvestation times, and so on. But then in the last century, around the 20th century, there were a lot of significant breakthroughs happened with the molecular biology, and the tools which are available for molecular biology they improved the understanding at the molecular level. So we were able to identify the DNA, RNA, and how they contribute to a plant cell growth and how they actually contribute to the crop production and so on. Further, the invention of recombinant DNA technologies also improved our ability to establish transgenic plants, which can have an improved ability to withstand all these kind of stresses. And in the current era, post 2000, I would say the ability to understand what's being encoded at the genome level, what the plant actually has as the hardwiring, if you can say, and then but not all the genes are always expressed, right? So there could be a difference in what's expressing in a leaf or in a root, how they are able to work in synchronization. Those can be realized by the use of large scale omics techniques. So you can try to look at the transcript level, so you can do the transcriptome analysis, or you can try to understand not all the transcript genes will eventually get translated. So you could look at the proteome level, and then not all the enzymes would have a sustained activity on the metabolites. So you could also look at the metabolome levels and so on. So integrated with the bioinformatic analysis, this gives you a lot of power to understand what happens within the rice. So we are currently in this post-genomic era. So what are the data? So as I mentioned, there are a lot of data available, right? So the rice genome was sequenced and it has subjected to multiple different genome annotations. I'm sorry, I'm not familiar with the current version and the current statistics of the number of genes and the chromosomes available. This could be improved based on the current state. And then even back then, there were a lot of transcriptome data was available. These were mainly based on microarray. I believe it should have been now more with the advanced techniques like sequencing based RNA-seq would have been available for characterizing what's happening in different stress conditions or what is happening in a transgenic plant, how plants actually rewire themselves to better adapt or not able to adapt. So we will be able to delineate both of these. And then there are also already at that point in time, about 10 years ago, there were a lot of proteome data sets were available. Out of the 30,000 genes which were annotated, the proteins were characterized for more than about 30% roughly. So this number would have also now increased by a significant amount. Last but not least, there are a lot of metabolomic studies which were also carried out in rice, which provided you the understanding of not only the primary metabolites, but also the secondary metabolites, the role of different secondary metabolites, particularly the terpenoids and their derivatives, how they are able to support the function of rice plants. So, in case of rice, the secondary metabolites may not be having a, a role which may be very specific to the health benefits, but rather it may have a role which has to play for the individual tissue development. Say, for example, how a leaf can be able to sustain the photosynthesis and what's the role of the secondary metabolites in the assembly of leaf tissue and so on. So the objective of this particular approach is to combine. So we already know all these data are available. So we want to combine all these things and then make sense of it in a holistic manner. So how we can do that? One way to do that is using systems biology approaches. So wherein you try to build computational models considering that these are the parts. So I can give an analogy of a car where if you have different parts of the car, then you may need to assemble them to put together so that they have a functional output which you can be able to ultimately drive that. So that's what systems biology does for you. So you will be able to assemble all these parts together, then you will be able to make some hypothesis which you can validate, and once you are able to validate these hypotheses, you will be able to use those models for testing further hypotheses. But then if these hypotheses have a mismatch with your experimental comparison, then you need to improve the data on which it's being built, and then you try to do this. So it's an iterative cycle in such way. So now going into a bit more detail of what we can do and what we have done earlier. So what we did at the initial stages, so in today's concept, today's lecture, I should have covered the different omics, including fluxome. So maybe you can try to consider this as a fluxome topic. What is a fluxome? So when you have different metabolites within the plant cells or any cells, how they are continuously getting transformed. So we know that a plant metabolic network or any metabolic network within cells, which actually carries out a series of enzymatic transformations. So these transformations can be captured as a snapshot, which is actually called as a flux. And when you take this as a holistic view, the whole cell level, then it is called as a fluxome. You are taking multiple fluxes at the same time in consideration. So in order to understand these fluxes, the one way to do is you can actually try to measure them intracellularly. So how we can do that? You can use a isotope label based approaches. So you will be able to understand how much of a glucose which is getting transformed into say G6P and so on in the glycolytic pathway. That's one way to do it. The other way is when you have a metabolic network built for the species of investigation, in this case it would be rice. If I know what is the different nutrients being consumed and what are the different products being produced and so on, then I would be able to use this information and try to estimate what is the transformation happening within the cell. So that's a computational approach. So we have actually used that particular approach in this next set of slides. So for that, we first need a metabolic network. So we used multiple plant resources, including plant metabolic network, Brenda, KEGG, Gramine, which is the genome resource and so on. And there was also a particular rice-specific metabolic pathway database present at that time called RiceSite. So we took the information from all these and then we performed a lot of manual refinements, for meaning that which particular reactions may happen only in plastic, which particular reactions may happen only in mitochondria, which particular reactions may have duplications across the network, and so on. So after doing this thorough manual refinement, then we were able to develop this model and subject it to a various in silico simulations, which were able to give us the intracellular fluxes. So at the outset, we established a generic rice model. Then we also set out based on the transcriptional regulation. As I mentioned earlier, not all the genes encoded will be expressed in all the tissues. So we used that particular logic to tailor our generic model, which encodes multiple genes. This is not a genome level model. So this does not take into account all the encoded genes, potential genes, but a subset of metabolic genes. Then we used regulatory rules to understand what are the genes among them are expressed, and then what would be conditionally specific. This enabled us to have a leaf specific metabolic network as well as a seed specific metabolic network. Why we did this? Because we wanted to characterize what's going on in a stress which is possibly happening at the drought conditions, whereas in the seed during the germination, if it is submerged, what happens during a flooding stress? When there is a oxygen deprivation, what happens? Okay, so first we want to understand. So I told you about we established these models, right? But are these models able to predict accurately the results? So that we wanted to first understand. So the collaborators, they were able to perform callus derived cell suspension cultures, and they made these cells to grow in different carbon sources, sucrose as well as glucose, and they tested their ability to grow both in aerobic and anaerobic conditions. So we use these data. So these are extracellular data. So it shows that the consumption of sugar, which is you could see a decrease in trend, as well as the increase in the cell mass. So by consuming the substrates, they were able to grow. And there were also some byproduct production, such as ethanol in case of anaerobic conditions. So using this information, then we were able to understand whether, so what we did in this case is, we used this consumption rate of the substrate, and then we allowed the model to predict the increase in the cell mass. And we were able to see that the prediction levels in almost all the conditions were actually quite within the reliable ranges of the model was able to predict quite accurately. That's what I'm trying to say. So with this confidence, then we were able to perform the conditions and estimate the intracellular fluxes. So you could see the different color intensity. What you see here is the flux intensity. So meaning that the transformation of G3P to 1,3-PGA is much higher than the transformation of DHAP to G3P. So how we were able to estimate this? We were able to estimate the values of the sucrose as well as the ethanol, the other byproducts which are coming out of the cells, and we were able to estimate what is going on inside the cells. So you could see that there are few key changes which is happening between an aerobic and anaerobic condition. The first thing we noted is that there's a significant increase in the net glycolytic flux. So it is comparatively very high, as well as most of them are channeled towards ethanol. Why this is happening? Because I'll come back to that later. Because in the anaerobic condition, there's a gross impairment of TCA cycle. So it is not able to respire completely through TCA cycle. Rather, in the aerobic conditions, there's a cyclic flux was observed in the TCA cycle. So because of this, it had to rely on the glycolytic fluxes, so producing most amount of ethanol, and it was not able to sustain the so that it can barely survive. So that's what the plants are trying to do. We also noted that in the case of anaerobic conditions, the breakdown of sucrose is slightly different from the aerobic conditions. So it relied on the PPDK and reduces the ATP demand, because sucrose consumption in plants, it requires ATP for the breakdown of the sucrose as well. So it tried to reduce by using an alternative enzyme which reduces the ATP demand as well. So further, this is a minor observation which also noted that the glycine is synthesized in the anaerobic conditions through GABA, which is not the case in aerobic conditions. Then we also try to use, so this is what we understood from the intracellular flux estimates, right? But we wanted to also confirm whether this is true. How much this is true in terms of what is happening in the cells. So how we can do that? One way to do is a comparison between the gene expression levels, which is measured both in aerobic conditions and anaerobic conditions. And then the other thing is we already simulated the fluxes in aerobic and anaerobic, right? So we can do a comparison between these two. Whatever agrees, then it will provide you a higher confidence that it is actually happening in the plant cells. Whereas if there is a contradiction, then you will be able to improve the prediction further. So as such, so we were able to, so whatever you see in the red or green is the actual validated enzymes which showed a change in the magnitude. Green means down regulation, red means up regulation, both found in the expression level as well as in the flux level. So this shows that there is a synchronized uh, activity both at the, because of the gene expression reduction, there may be a possibility that the flux levels are also decreased. But then there are also some contradictions which we observed, which are denoted in the blue, which showed the opposite direction. Probably the flux showed a reduction, whereas the gene expression showed increased expression. Okay. So the next bit, what I wanted to focus on in this work is, so we understood a lot of different fluxes, right? So, or the reactions which had a difference between, say, aerobic and anaerobic. But what do they mean? How they are regulated? So we wanted to understand that. So how we could do that? We identified the set of reactions which showed a same directional change, be it aerobic to anaerobic, being upregulated or downregulated. Then we looked at the potential transcription factors of these genes and wanted to understand how they are regulated. So this analysis actually showed that the multiple transcription factors, a set of MIB, VZIP, zinc finger, and ERF transcription factors, which is a classical plant transcription factors, and they had a significant role in the combined switching of these enzymes, particularly the alcohol dehydrogenase, aldehyde dehydrogenase, pyruvate dehydrogenase, and the sucrose synthase. They work in a synonymous way. How? Because of the shared control of individual transcription factors, as you could see here. So when we look at the motifs of these individual genes at the promoter regions, you could see that they are being controlled in a fine, refined manner. So this suggests that the adaptation happens at the transcriptional control level, which is realized in the flux level. So that's what is the ultimate story here. Then moving on, I also mentioned that we established a leaf model, right? So we wanted to also evaluate what's going on in leaf when there is an increase in the kind of drought stress. So this shows the carboxylation to oxidation ratio of Rubisco, which I feel that most of you would be familiar, that Rubisco will not be able to take all the carbon dioxide on its own. It will also take oxygen as well. This happens, and then when there is an increase in the stress, the ratio becomes much higher as well. Like the amount of oxygen being utilized, it becomes very high. In that sense, the carbon dioxide fixation levels, they drop down, and then you see a steep decline in the performance of the plants. So we simulated different ratios. What is happening at the stress level? The ratio will become very low, and we wanted to investigate what's the change happening in the entire flux level. So this shows the Calvin cycle within the plastid, and you could see that the Calvin cycle almost have no difference depending on how much of carbon dioxide to the oxygen ratio available. But what happens is, as the oxygen increases, the bypass in the photorespiration, which involves both the plastid as well as the mitochondrion, they have an increased flux. Because of this, they also produce a lot of reactive oxygen species. So this actually shows that decreasing the CO2 also decreases the cell ability of the cells to grow. That is what happens in the drought stress. And then this increased flux in the photorespiratory pathway, which this particular set of reactions actually reduces the flux in the TCA cycle. So this shows that how the plant is not able to channel all its nutrients in the unfavorable conditions in the drought stress. Further, we also evaluate. So we in this step we noted that multiple reactions are involved, right? So we wanted to see if we can do a transgenic plant which can get rid of some of these photorespiratory pathways. Can they survive? But it showed that the simulation showed that in fact all the reactions in the photorespiratory pathway were actually important. So how we did this? We were able to computationally remove each of these genes and then analyzed whether plants are able to still photosynthesize. When you remove any of this, the plants were immediately dying. They were not able to simulate any photosynthesis. Why this is happening? Because as I mentioned earlier, Rubisco will not be able to completely utilize everything into carbon dioxide. So that is why the ability of C4 plants, which has a different morphology, is able to concentrate better the CO2 within the bundle sheet. So then, yeah, this just indicates that all the photorespiration genes are essential. So you have no chance to remove any of this. So you have to live with the bypass of the photorespiration. Okay, so we got some insights from these rice plants in the smaller model, right? But it was not able to provide any information about the secondary metabolites, which is a large family in plants in general. So because of that reason, we wanted to establish a genome level metabolic network to understand the secondary metabolic pathways particularly, and how different light or other stresses is able to control the transcription of different pathways. So for this, first we built a genome scale model of rice. So this shows the plastid, and maybe this is a mitochondria, ER, and so on. Different colored nodes indicate the different pathways. So this shows that plant has a very big metabolic component within the plastid itself. So which is almost equivalent to what you find in cytosol. So they have a very critical role in plant metabolism. They also have duplicates, multiple pathways like glycolysis, lipid metabolism, and several other pathways. They have a duplicated version in the plastid as well. And then this shows the number of gene associated reactions. But one major update here, what we did is that when you have different colored lights, which can be segmented based on wavelengths, how the photosynthesis actually happens, the photon absorption happens, and how much of energy is actually derived in terms of proton. So we did this using earlier work which was done on a photosynthesizing algae. So what we did is plants were able to absorb the light in the wavelength of 400 to 700 nanometers. So we were able to break down the photons which are incidental to plant in this category. Whatever falls under the 400 to 430 and so on, 430 to 450, we segregated them and we were able to get this. So why we have to do this? Because when you have a change in the color of the light, the plants will be able to have only a set of photons and their efficiency actually varies. So in this sense, we were able to show that what is being incidental and what is also not able to absorb. So there is a whatever is incidental will not be absorbed. Some will have a very high absorption, some will not be able to have a very high absorption. Why this is critical? Because we also showed that across different wavelengths, when you have the photosynthesis, the amount of photosynthesis happening actually changes. So at the blue light as well as at the red light, it has the maximum amount of photosynthesis happening. So with the model, you could see the dotted lines is the experimental, whereas the solid lines are the model simulated ones. So it was able to grossly reproduce the trends. So the ultimate usage of this model will be shown in the later slides. So what we did is, some of our collaborators, they were able to grow the rice plants in different colored lights in a conditioned environment. So we did this in red, blue, which showed the maximum amount of photosynthesis, also green and white, which has a mixture of all lights, and then complete darkness, no light at all. They are not exposed to any light. And then we captured their metabolome, what is happening, what is the amount of different amount of metabolites within this plants, what is the different gene expression changes happening in this. And then we already have the metabolic network, and we did a series of analysis which had potential applications in the circuit design, transgenic plant, as well as the greenhouse design, as I mentioned earlier. So first, let's look at the outcome. So when we noted that the plants grown in different colored lights, they had different outcomes. Obviously, the ones which are on dark doesn't have any light. They were actually pale in color and they were actually malnutrition because obviously because they cannot photosynthesize, they were ultimately relying on the oxidative metabolism. But then interestingly, we know that blue and red could be very favorable for plants, right? But plants grown in blue, they were actually stunted. They were not able to grow uh, taller. Whereas plants grown in red did not show much difference in white, which is the natural sunlight resembling the sunlight. Green also had a similar kind of distinct phenotype, but the shoot sizes were actually changing. So, in order to understand this, we also measured the intracellular secondary metabolite concentrations. Particularly, we focused on the terpenoids, carotenoids. Different type of carotenoids they actually varied. For example, blue had the maximum accumulation of carotenoids, and red had the least amount of carotenoids. So, I'll tell you why this have the differentiation later in the later slides. So the first thing what we wanted to understand is at the global level, how much of the gene expression changes. So you could see that R denoting red, blue, green and dark, and W denoting white, red, blue and green. So when you look at this, red is very close to white in terms of gene expression. The correlation levels are very high, whereas it is far away from dark. So this is a PCA plot. The closer the clustering, it indicates the similar they are. So white and green, they were almost very similar, and red is bit farther from them, and blue is also a bit farther from them. Whereas red, the dark is completely different from the other colored growth. So within the light treatments, blue and red, they have a very distinct outcomes in terms of gene expression. So they change the gene expression in a fine-tuned manner. So what are the pathways which have an effect when there is a change in the gene expression? So that's this plot indicates that. So in blue, we noted that the secondary metabolism, particularly terpenoid biosynthesis, have a very high amount of gene expression. And also the photosynthesis was the highest in blue color. So they were able to fix more CO2 and they were able to dedicate and channelize most of them into terpenoid biosynthesis. On the other hand, red had the least amount of photosynthesis when compared to the green, blue and the white, not the dark. And then they also had a relatively lower amount of allocation into the terpenoid synthesis and other secondary metabolite synthesis. In terms of dark, there is a elevated amount of degradation of certain amino acids, and there is a complete reliance on oxidative phosphorylation, and several amino acids were also started to degrade. And in green, it has a more balanced growth with increased amount of the plant hormone synthesis like IAA, indole acetic acid, and so on. So this plot shows that we were able to map what are the changes happening in the pathways, different pathways, and whether they are upregulated or downregulated when compared to the white light. So you could see that major change is happening in red and in the dark. So what are the reactions or the pathways which is having a major difference? We noted that majority of the secondary metabolite like the plant hormone like gibberellin, jasmonate and abscissicate metabolism, they were very different in dark. They were almost non-existent in dark, and they were upregulated. Some of them were upregulated in red and some of them were upregulated in blue. So this probably indicates that the combined role of your secondary metabolite synthesis and the plant hormone synthesis probably have a different outcome. Blue may favor more on the carotenoid biosynthesis, which may contribute to the smaller plant size, whereas the red may have a contribution towards more on plant hormone synthesis, which may have a role to play in the taller plants. Then again, we wanted to understand whether these encode for any common set of transcription factors. So we looked at the genes and the common motifs, and we were trying to pick out what are the transcription factors possibly controlling them. So we indeed identified a set of transcription factors which could be color specific. Sorry. So you could see that here we noted that a set of MIB genes, including several like BZIP, WRKI, and zinc finger, they could be blue specific because among the genes which are enriched in blue, we were able to pick out only these set of transcription factors. Whereas MIB was particularly identified to be a red specific. When we look at the genes which are increased in expression in red, they were mostly regulated by MIB. So this transcriptional factor difference will also play a role in how the plants reflect to different kind of lights. And then we also made a comparative analysis of what we identified and what was already reported in the literature. And we also noted that some of the identifications in the blue specific light were actually contradicting as well. So it's not 100% foolproof, this kind of analysis. The last analysis what we did is, as I mentioned earlier, we also did the metabolome comparison in both the red and blue lights. So what we did is we looked at what are the metabolites which are elevated in blue as well as in red. This showed that the sugars, primary sugars such as fructose, glucose, and even sucrose, there was not much of a difference. But trypsin, they were actually higher in red. But on the other hand, amino acid metabolism and other carotenoid metabolites, they were higher in the blue color. So this may indicate that whatever being synthesized, blue color will try to produce more and more mass, which may contribute to the plant reproduction. Whereas the red light could possibly store them into a sugar moieties rather than reproducing them into a newer plant cell and so on. So that is the hypothesis which we can get out of this. So in summary, this set of data, what we were able to do is we were able to integratively analyze different kind of omics data, and this suggested us what are the key transcriptional and metabolic adaptations happen in flooding as well as the drought stresses, which I showed earlier. And we also were able to establish the finer changes which happens when you grow them in different colored lights or when there is a gradual change over the wavelengths. So the potential applications of this is when you are able to characterize what is happening in the photorespiratory pathway or the fermentation adaptation, you will be able to better establish transgenic plants, or you will be able to better design a better greenhouse effect and so on. Okay, now I want to move on to the second set of the second set in my lecture. So, which is the Korean ginseng. Korean ginseng is actually a herbal extract which comes from the roots mainly. So, it's a plant which is mostly found in the hilly regions of Korea, South Korea as well as North Korea, and traditionally Koreans they have been consuming this ginseng for various health benefits. So the traditional medicine of Korean literature, it also documented a lot of health benefits. The particular health benefits which I am familiar of is some of its anti-cancer effects as well as the neuroprotective effects. So it is known to be one of a very favorable stress reliever. So when people there are also multiple studies which have showed that people, when they were able to consume it in a day-to-day basis, they had a better neuroprotective effect. And then it has also had to show that it has a higher amounts of antioxidants, as well as it has a role in angiogenesis as well as the cardiovascular diseases. So because of this reason, they consume ginseng widely in Korea in different forms, right from tea, jellies, extracted oils. I think even they have things like chocolates, multiple different varieties they try to consume it, or even formulated capsules as well. So because of this reason, ginseng they grown into prominence. What makes ginseng the health beneficial effect it has? It's mainly because of a second group of secondary metabolites called ginsenosides. They are derived from the obviously the MEP pathway as well as the MVA pathway, with the intermediates as IPP and DMAPP. And these are the derivatives, the terpenoids are then made into triterpenoids. Building blocks are just kept on adding, and then they are glycosylated. So this glycosylated triterpenoid saponins, they are ginsenosides, and there are three major categories: RB, RG, and RO. Their levels actually vary in the different parts of the plant, the leaves, shoots, and roots. So that requires you to understand why this is happening, as well as which is the most relevant issue to look at. One of the key challenge in ginseng research at that time was the genome sequence was not available. So because of that reason, we wanted to focus on the sequencing of ginseng plant so that we will be able to better understand the biosynthetic pathways of these as well as their regulation in which tissues, how they are regulated and so on. So we used an approach where we first sequenced the long we used a PacBio based approach, which is a long reads as well as a short reads. So it's a combination of both Illumina and PacBio. And from the raw data, we were able to do the quality control and we were able to identify and align the sequences to eliminate duplicates. And then we were able to come up with the de novo assembly. From this, we were then trying to reduce the number of scaffolds, or in case of chromosomes, if you could, the level which is above these chromosomes is the scaffolds. If you are not able to place it to the chromosome level. So in this, we were not able to place at the chromosome level. So what we finally got is a scaffold level. So with this, we were able to then annotate what is happening in the ginseng. So the first thing what we notice, so this table gives you some kind of snapshot on the assembly. What is the total genome sequence? The number of scaffolds was still around 10,000 scaffolds. So as and when there is an improvement in the genome assembly, this gets drastically reduced and it gets into some of the chromosome levels. Total number of genes, we identified a very unexpected number of around 60,000 genes. When compared to rice, which is only 30,000, this is a very high number. So initially we were suspecting whether it is probably a misannotation, but then we used multiple confirmatory methods and it was indeed confirmed that there's a possibility of 60,000 genes. Most of them were actually duplicated. And it is actually a farthest derivative from carrots among the known plants. So when we compared about the multiple genome sequences we had for other plants, we were able to relate it and trace back to carrots. And the genome synteny, the plot what you see here is the base pair level similarity among multiple scaffolds. So each line shows a scaffold in the Panax ginseng, and this shows a chromosome in the carrot assembly. You could see that they share a very good synteny, meaning that similarity between the base pair level. And there are also chromosome level, the intermixing which happened in the chromosomal level. As I mentioned, most of the ginseng genome is duplicated. So multiple chromosomes or the scaffolds, they have the same kind of coding sequences or even the non-coding sequences. So that is what is shown in the last plot. Then coming into the major aim of the study. So we were able to completely deconstruct how the ginsenoside synthesis happens. So starting from the MVA and MEP pathway, which happens in the plastid and the cytosol, it actually produces the plant sterol, which is squalene, and oxidosqualene, which is usually produced in all the plants. But then the branching actually starts after that. The RO type of the ginsenosides, they branch out from here, whereas from the damarine type of the ginsenosides, they start from the branching here. So the oxidosqualene is here, and then it also has other minor ginsenoside varieties. And on the right hand side, what you see is the similarity of the genes which are from the closest ancestor from carrot, including the Panax ginseng and so on. Even tomato is also provided there. So this is the secondary, but they do not have the glycosyl transferases. So those pathways actually they stop here. Whereas the dots which you see in the orange color is because of the glycosylation which is happening to the terpenes. And the heat map what you see there is the expression levels of multiple genes. The top is the FFPPS, squalene synthase, squalene epoxidase, and so on. And then the levels actually varies from the shoot parts as well as from the root parts. That is the one comparison we made. And the other comparison is the different cultivars. So within ginseng, there are multiple types, subtypes. So they also had a different variation in their expression levels. Some of them, they had a high amount of expression, whereas some other they do not have that level of expression. Then going on further with the comparative analysis of previously sequenced plants, what we also able to look at is the similarity between multiple other plants. So all shown here are dicot plants, and among them they had about 1697 ortholog gene clusters. So meaning each gene cluster may have multiple genes. So they were grouped into clusters and then they were able to be compared across different plants. So you could see that most of this similarity is again shared with the carrot. The part here, which is, you could see that more than 2150 is shared with the carrot. Whereas if you look at the similarity between ginseng and Arabidopsis is much lower. There is only 110 gene clusters are shared. So this shows that, this again confirms that it has actually evolved from carrot, and it may have a common ancestry. And this shows the functional evolutionary. What does that mean? So I showed in the previous different gene clusters, right? So what are the gene clusters they try to encode? What are the different genes they encode? What are their functions? So you could see that most of the differences found in the ginseng is the things which are specific to chromatin binding, endoplasmic reticulum. Why we see endoplasmic reticulum and carbohydrate metabolism? Because the ginsennoids are glycosylated terpenoids, so which requires the glycosylation to happen across the different organelles. So that's why you also see the organelle specific differentiation as well as the carbohydrate, which provides the precursors for glycosylation. So you need glucose molecules and different other carbohydrate molecules for the glycosylation process. So those are the major difference which are in the ginseng when compared to the other crop plants. So eventually we also had a chance to look at, so we did not go into the detail of this in this work. So the collaborators also had the gene expression profile at cold, salt stress, drought stress and heat stress. Cold should not be considered as a stress in case of ginseng, because as I mentioned, it is preferred to grow in a hilly region. So because of that, certain amount of ginsenocytes, they were known to be enriched in the cold regions when they are grown in the cold, and when they are avoided from the sunlight. So if they are exposed to a direct sunlight, the proportion of ginsenocytes are also varying. So when we look at the different type of gene expression which were happening, particularly in the cold, we noted that the ability to express different type of fatty acid desaturase as well as the cytochrome P450, which is a monochrome oxygenase, these levels were completely different. So that may be giving them the flavor of the correct proportion of different ginsenocytes. So that is the major difference what we observed in a cold when compared to the other conditions. Okay, so in summary, we identified few things in this work. One is we were able to establish the genome size of ginseng, which is 2.98 GBP. But the interesting thing is it encoded a way higher number of genes when compared to the closest ancestor, which is carrot. And then we also noted that it would have occurred because of two whole genome duplication effects from the carrot, and it may have occurred within this particular species level, which is Araliacid. And then the Dameron type of ginsenocytes is only specific to Panax, which were not found in other type of species, and they are largely produced in shoot tissues rather than produced directly in root. How we were able to ascertain this is based on the gene expression of the multiple tissues as well as the different metabolite levels, which we were able to do that. And then, as I explained in the previous slide, why they were able to do the freezing tolerance and the different type of the ginsenocytes, what we see in the hilly regions is mainly because of the fatty acid desaturase levels of expression. We also were able to establish a metabolic network which focuses mainly on the ginsenocyte biosynthesis, which will provide opportunities for further expressing them probably in other microbial cultures as well. So if you want to produce ginsenocytes in microbial cultures, now we will be able to know like what are the genes you need to clone and express in microbial cells. With that, I come to the end. I will be able to answer any questions. Thank you.